Hello everyone. This lecture comes from Essential Biochemistry, Chapter 4, Section 2, and we also cover a little bit about collagen in Section 5.3. So today we talk about proteins, and why are proteins biologically important? Well, they're involved in cellular function, such as enzymes, which are the catalysts of the cell. They're involved in storage and transport. They have a structural role and mechanical support. They're involved in information and communication. And the reason that these are so important is shown here. But what we want to do is we want to approach our studies using a structure-function relationship. It's really important to understand protein structure so we understand how they work. So before biochemists understood how proteins were put together, what they actually looked like, it was difficult to discern their function. So that's something that we will be able to do now that there is so much known about protein structure. So first of all, how do we make a protein? Well, we make a protein from amino acids. So we have two amino acids right here, and they're drawn so that their amino groups are on the left and the carboxyl groups are on the right. And what we're going to do is we're going to involve the carboxylate group on this amino acid on the left and the amino group of this amino acid on the right. And shown in red is the water that we're going to form when we do this condensation reaction. We're going to lose this oxygen and these two protons and form water. And now this carbonyl group right here will be bonded to the nitrogen. And what we have when we lose our water is the carbonyl group, the carbon bonded to the nitrogen, and this is an amide bond. You might remember that from organic chemistry. But we're going to call it a peptide bond because it's a special amide bond because it involves the linkage of two amino acids. So when we do this, we still have the amino terminus on the amino acid that was on the left. So we're going to call that the N terminus or the N terminal or the amino terminal. And on the right, we have the C terminus, so that's the carboxylate group that's on the right side. And conventionally, when we draw protein structure, the N terminus is on the left and the C terminus is on the right. So what this means is that when we looked at amino acids, we saw that the amino groups were positively charged and the carboxylate groups were negatively charged. So notice when we put these amino acids together as we start to build our protein structure, that this negative charge on this carboxylate group and that positive charge on this amino group they're gone when you form the peptide bond. So that means that the only places where you have charges now will be the N terminus, the C terminus, and the R groups if they are the R groups that have the potential to be positively charged. Remember, not all of them do. So to illustrate, you should do an exercise for yourself. So here I've shown free amino acids. I've shown a methionine, an aspartate, a leucine, and a tyrosine. So you want to form a tetrapeptide where you're going to link all four of these together. So what you should do now is you should stop the video and you should draw the structure of this tetrapeptide. And then when you're finished, you should restart the video. Well, hopefully, when you drew your structure, this is what you got. So here we have the methionine, the aspartate, the leucine, and the tyrosine. And here we can see that we have the peptide bonds, and the R groups form side chains. Now, if we want to name this thing, we could, because it only has 4 amino acids, we could call it methionine, aspartyl, leucine, tyrosine, or we could call it met-aspalutyre, or we can use the one-letter code MDLY. Either one of those would work, but you can see that the one-letter code is much more compact. But the other thing that we should notice again is that we have a charge on the N terminal, the NH3 plus group over here on the left, the negatively charged carboxylate group on the right. The only side chain that is charged is the aspartate. The methionine, the leucine, and the tyrosine aren't charged. So here, notice that we only have 3 functional groups that are charged. So we put together 4 amino acids to make a tetrapeptide. So usually we say peptide if maybe you have between 3 and 10 amino acids, maybe a polypeptide up to 40 and a protein 40 or more. These are not hard and fast rules. Sometimes it depends on what book you pick up. But the vast majority of proteins that we know about are between 100 and 1,000 residues. Usually you need about 40 for proper folding. So what we should appreciate is that the amino acid composition influences the solubility and ionic character of the protein. Just like the 26 letters that we use to make words in our alphabet, the 20 amino acids that we choose will influence the character of the protein. So we want to talk about protein structure, but before we do that, we're going to talk about the peptide group. So I showed you how to form peptide bonds. But we want to talk about the peptide group because there are various levels of what we call protein architecture. But before we talk about what this means, we have to define the peptide group. The peptide group consists of the peptide bond and the 2 alpha carbons on either side of it. So if we look at the peptide bond, so here's the peptide group. So here's our peptide bond, and here are the alpha carbons on either side of it. So what I want to appreciate right here is that this is an amide group right here, and you might learn from organic chemistry that you can draw resonance structure from this. If you swing these electrons over here to form a double bond and swing these electrons over here, you'll have a double bond between the carbon and the nitrogen, a positive charge on the nitrogen and a negative charge on the oxygen. You should also remember from organic chemistry that the actual structure isn't a fluctuation between these two, but it's a mixture or a hybrid between the two. So what this means is that this peptide bond that we typically draw as a single bond between the carbon and the nitrogen, what we should remember is that it has partial double bond character. So it is shorter than a typical single bond. The carbon-oxygen bond that we typically draw as a double bond has maybe between single and double bond character. So it's longer than a typical double bond. Both bonds are polar because of the electronegativity differences between carbon and oxygen and between carbon and nitrogen. We can see that we have these groups can be involved in hydrogen bonding where the NH can act as a hydrogen bond donor and this oxygen can act as a hydrogen bond acceptor. And we should also note that because we have this double bond character, the shape of the peptide bond is planar and the carbon and the nitrogen are sp2 hybridized. So this is an example of what it looks like. Again, this is the peptide group. We have the peptide bond, and here this carbon is shown in gray. The carbons on either side are shown in black. So what we've done here is shaded the peptide group in blue. So again, this is drawn with the sp2 hybridization, and the and because we have a partial double bond character, if we look at this carbon-nitrogen bond right here, we can draw this as trans or cis. So I hope you can tell from this picture that nearly all peptide bonds are trans. You can see that you have a lot less steric hindrance with the carbon-nitrogen bond in the trans conformation and then the cis conformation. So the exception is peptide bonds involving proline. Remember, proline is the only secondary amino acid. So about 10% of those are in the cis conformation. So here is a drawing of a peptide bond, actually 2 peptide groups. There's a peptide bond here at the top and another one here at the bottom. And again, shaded right here are 2 peptide groups. So if I'm looking at these peptide bonds right here, the peptide bonds can't rotate because they have partial double bond character, but the bonds on either side of them can. So we refer to this bond right here. So this is involving the nitrogen and the adjacent alpha carbon, and this involves the, this is the nitrogen and the adjacent alpha carbon right here. So that is referred to as psi, and over here we have the carbon in the peptide bond and the adjacent alpha carbon. So we're going to refer to this bond as psi. And both of those bonds are single bonds, so they can rotate. So theoretically, you can have several conformations. So you can't rotate the peptide bond, but these, the carbon alpha nitrogen bond and the carbon alpha carbon bond, are single bonds, and you can. So we can plot something called a Ramachandran diagram, which I'm going to show in the next plot. But I want you to appreciate that we can't rotate these single bonds, even though they are rotatable infinitely, because we could get some of these atoms closer than their van der Waals distance will allow us. We can't have these overlapping electron clouds because electrons are negative and they would repel each other. And we should appreciate that the electron clouds are larger than these space-filling models show us. So if we have a Ramachandran diagram right here, we can show the rotation of the bond that we defined as the psi over here on the y-axis and phi on the x-axis. And this diagram shows us sterically allowed and sterically hindered conformations. So these blue shaded areas right here are sterically allowed. The green are sterically allowed, but it's a little bit more crowded. Here we see sterically allowed conformations that we call beta sheets. I'll show you that in a minute. And these are alpha helices over here. So what we find, not surprisingly, is that proline is the most conformationally restricted amino acid, and glycine is much less sterically hindered than the other amino acid. The other thing that you should appreciate is that there are far more forbidden rotational angles than allowed rotational angles. So this shows the extended conformation of a polypeptide. So if we look at this from the amino terminal on the left to the carboxyl terminal on the right, and we can see all of our peptide bonds, the peptide groups are shaded and shown here as their planar geometry, or these are shaded right here. And in white, we see the R groups. So we have a main chain, and then we have the side chain. So the main chain, or the backbone, contains the peptide groups, and the amino acid side chains project out from the main chain. Another way to show this, maybe more simply, is this gray tube right here contains all the peptide groups, and then these little bumps right here are the amino acid side chains. So this is the tetrapeptide that we drew in the beginning of this talk. So you would think that you could form lots of different conformations even with these limitations, but proteins usually assume only one conformation, which is the native conformation. And that's not to say that proteins are rigid, because they're not. They do have some ability to breathe, but we will refer to the accepted conformation, the biologically active conformation, as the native conformation. So these 4 levels of protein architecture that we want to talk about today. So the primary level is the sequence of the amino acids in the protein. And some proteins have disulfide bonds, and if they do, we would have to indicate the position of the disulfide bonds if they have them. So what we want to talk about now are secondary, tertiary, and quaternary structures. So again, the primary structure is the first level of protein architecture. It's defined as the sequence of amino acid residues. This is determined by the DNA sequence. So this is insulin right here. So insulin consists of 2 chains. We'll call this an A chain and a B chain. And they're linked together by these disulfide bonds right here. Between the chains, we have another disulfide bond within the same A chain. Now this doesn't tell us anything at all about what insulin really looks like. It just tells us that we have an A chain and a B chain. It shows us the sequence of the amino acids in each of these chains, and it shows us the positions of the disulfide bonds. We should note, although this is sort of a generalization, that typically we find disulfide bonds in extracellular or oxidizing conditions, whereas the inside of the cell tends to be more in a reduced form, so we are less likely to find disulfide bonds there, although that is a generalization. So what's this next level, secondary structure? So how do I define that? Well, it's the local spatial arrangement of the polypeptide backbone atoms with regard to the conformation of its side chains. And it refers to any regular repeating structural motif that has residues with these repeating phi and psi values. So that what does all of that mean? Well, it means that we have these sort of regular repeating structures, and if we look at proteins, we can see that we can predict theoretically, and that's what the early biochemists did, is they predicted what kinds of structures they would form, and then they used X-ray crystallography to see if those structures were actually there. So typical examples of secondary structure are alpha helices and beta sheets. So when the polypeptide chain folds, it wants to satisfy as many hydrogen bond requirements as possible while minimizing steric strain. So let's look at the structure of the alpha helix and the beta sheet. So the alpha helix looks kind of like a corkscrew. It is right-handed, which means if you held your right hand and you pointed your thumb up, that the direction that your fingers curl would be in the right-handed form. The other thing you can do is look at this right-handed ribbon right here and imagine you put a pole down the middle of it, and the ribbon crosses in front of the pole and it points to the right. So that's a right-handed helix. What we will find that holds this this alpha helix together is that there will be hydrogen bonds between the carbonyl oxygen of amino acid N to the alpha amino nitrogen of amino acid N+4, and I have a video that shows this pretty well. This ribbon right here just shows the polypeptide chain or the main chain. There are no R groups shown in this diagram. The other thing that we should note is just because we're showing the ribbon, we're not showing the space-filling model, we're just showing the ball and stick. It makes it look like there's lots of empty space in the alpha helix, and it's not. The core of the alpha helix is very tightly packed. So let us look at the helix animation. And so what this video will do for us, it will count amino acid for us. So, so again, here we have amino acid 1, 2, 3, 4, 5. So this is amino acid 1. That's amino acid N+4, or amino acid 5. So you're going to go 1, 2, 3, 4, 5, and again it's going to show the hydrogen bond between the carbonyl group of the nth amino acid and the and the NH group of the N+4 amino acid. There will be, if you have 18 amino acid residues, it will make about 5 complete turns, which works out to 3.6 amino acids per turn. All right, so we will get out of there and go back to our PowerPoint. Let's look at another example of secondary structure, which is the beta sheet. So this occurs between neighboring polypeptide chains rather than just one. The alpha helix, it just involves the structure of one of a single chain. So proteins can fold so that you have polypeptide chains that are next to each other, so they are neighbors. And these will be held together by hydrogen bonds. They can be parallel or anti-parallel. So this shows a structure of a beta sheet, which is shown in green, and the amino acid side chains, the R groups, again project out. This picture right here shows 2 neighboring polypeptide chains that form a beta sheet, and in this drawing the R groups are not shown. So this shows how we could have anti-parallel or parallel beta sheet. So if we go in the directions of the arrows, we're going amino to carboxyl direction. If we go amino to carboxyl direction in this direction, and then we loop around to this direction, this is anti-parallel because we're going amino to carboxyl here and amino to carboxyl here. This is parallel. We go to amino to carboxyl here, and we have a big long loop here, and amino to carboxyl here. So where the arrows are meant to represent beta sheets, and you'll see hydrogen bonds between these 2 neighboring chains. And that's shown here. So there's also an animation that we can go to. So again, this is one of the flash animations that I rescued. So this shows an anti-parallel beta sheet. And we're going to go in the amino to carboxyl direction here and the amino carboxyl direction here. And here you can see how the hydrogen bonds in the neighboring chains form. And they're sort of straight on, whereas if you look at the parallel ones, there are still those hydrogen bonds, but they're a little bit at an angle. So you can see here the hydrogen bonds are being highlighted in yellow. So both of them are formed by these completely extended strands. So here we have 2 residues per turn and a pitch of 7.2 angstroms. So that went maybe a little bit too fast. Okay, let's go back to our PowerPoint. Actually, let me go back to this. So again, we have the amino to carboxyl in this direction and the amino to carboxyl in that direction. That's an anti-parallel beta sheet, and you can see the hydrogen bonds are kind of straight on. Whereas we go to amino to carboxyl here, there would be some other kind of loop that they didn't show that takes us to amino to carboxyl here. We still have those hydrogen bonds, but they are not straight on. So one of the things that we should note is that Linus Pauling and Robert Corey were X-ray crystallographers at Caltech, and they worked out the rules of structural chemistry, including the planar peptide group, that was consistent with protein structure. So they proposed that these alpha helices and beta sheets could exist before the X-ray crystallographers actually were able to experimentally verify them. And they tend to get a lot of attention. Herman Branson doesn't get as much attention, but he played a pivotal role. He was a physicist on leave from Howard University. He was doing a sabbatical at Caltech, and he did the calculations that determined that the alpha helix and the beta sheet were consistent with the structural chemistry. Because over in England there was another group of scientists working on the structure of hemoglobin, and they did not have a Herman Branson to help them work out the calculations, and they were working on myoglobin and hemoglobin, which have lots of alpha helices. They should have discovered the alpha helix, but they didn't. So Herman Branson was really pivotal to Linus Pauling and Robert Corey being able to propose that these structures existed, and then for the X-ray crystallographers to verify that they did. So we're going to stop there with our primary and secondary structure. We do want to talk about tertiary and quaternary structure, but we will do that at a later time. What I'm going to do now is I'm going to talk about collagen, because collagen is an example of a fibrous protein, and it is fibrous proteins are water-insoluble, physically tough proteins that provide mechanical support. They have an amino acid sequence that's repetitive and regular, and they're usually dominated by a single type of secondary structure. So they're good examples for illustrating secondary structure. So what are the characteristics of collagen? It's the most abundant protein in vertebrates. It forms strong and soluble fibers. It's a component of connective tissue such as bone, teeth, cartilage, tendons, skin, and blood vessels. There are more than 16 different types, but in humans usually you have 3 types that predominate. So starting with the primary structure, there is a pattern. So this diagram right here shows, has purposely drawn this so we can see this. We have glypromet, glyprosir, glyproarg. So every third amino acid is glycine. So so we can say glyXY. X is often proline, and Y is often hydroxyproline. So we'll use hype to identify hydroxyproline. That's not one of the 20. It's an amino acid that is hydroxylated. Proline is hydroxylated after the protein is translated. And less often you might have hydroxylysine. So let's look at this. So our proline could be hydroxylated on carbon number 4 or carbon number 3. The lysine could be hydroxylated on carbon number 5 of the lysine. So again, these aren't the standard amino acids. You have to make the protein first, and then you will hydroxylate these certain amino acid residues. So the collagen secondary structure, the collagen forms a helix, but it's not an alpha helix. It's a left-handed helix. Remember, alpha helices are right-handed. And also note that the alpha helix had to be put together by those hydrogen bonds that I showed you in the video. You don't need that in a collagen helix because you have the proline geometry that forces the turn. So if you have this gly-pro-pro, this secondary structure of the proline will help direct the structure of the left-handed helix. So what we get are 3 amino acids per turn. So just to convince you that the collagen helix and the alpha helix aren't the same, collagen has 3 amino acids per turn, whereas the alpha helix had 3.6 amino acids per turn. You don't typically find proline in an alpha helix because its geometry doesn't allow it to fit, whereas proline is absolutely essential to the formation of the collagen helix. The collagen helix is left-handed, the alpha helix is right-handed. You need those intramolecular hydrogen bonds to keep the alpha helix, that corkscrew structure, together, but you don't need it for the collagen helix because the proline geometry will form the structure for you. So this is what a collagen triple helix looks like. So we're going to start with one of these chains, and then we're going to, that's the space filling model, and then we're going to put 3 of them together. So here's a space filling model right here shown in part B, and if we take 3 of those chains and put them together, we have a triple helix. So the collagen, the individual chains are left-handed, but when you put this together, you have a right-handed triple helix. So that means you don't pull out the twist because the way that the strands of your coiled rope, the strands are coiled in an opposite direction than the actual rope itself. And here's another structure right here that shows how the triple helix comes together. So if I were to stand at the top of the left-handed collagen helix and look down, I'd see that in my 3 amino acids per turn, remember every third amino acid was glycine. So what that means is that when you make the 3 amino acids per turn, the gly-pro-pro, the gly ends up in the center, and when you put these 3 chains together, you have glycines in the center of the triple helix. If you remember your amino acid structure, glycines, our group just consists of 1 hydrogen. It's the smallest amino acid, it's the only one that's going to fit. So that's why it's essential that glycine is every third amino acid and that there are 3 amino acids per turn so that the glycine can pass through the center of the triple helix when the triple helix forms. You can see what happens if you don't. This is a mutant collagen right here. So where you see that yellow, those are alanines. So if there is a mutation in the genetic code, you'll get your 3 helices, and that means you'll have an alanine at each point of the 3 strands that make this come together. So you can see that here we have the 3 chains packed very tightly together, but alanine, which is not that much bigger than glycine, you can see that the shape is deformed and there's a distortion of the structure, and it's not packed as tightly together. And that's going to cause problems for the kinds of the body structures that contain collagen. So let's look at this proline residue. So I showed you that the proline was hydroxylated. There is a reaction that occurs after you make the protein. So here's the structure of the protein. Here's the proline. You need oxygen, alpha-ketoglutarate. You need an enzyme called prolyl hydroxylase, and you'll need ascorbate, which is vitamin C. And one of the oxygens in the molecular oxygen ends up on the oxygen of the proline. The ascorbic acid is vitamin C. Vitamin C is needed because the prolyl hydroxide has an iron in its active site, and the iron needs to be in the +2 oxidation state. If it's in the +3 oxidation state, it doesn't work. This enzyme also has to hydroxylate proline as part of the protein chain. If you just had free proline, that does not work. I wanted to tell you, because usually we cover this around September 19th, is that is International Talk Like a Pirate Day. So we'll figure out in a second why we are interested in International Talk Like a Pirate Day when we're talking about collagen. And the reason for that is that if you have collagen without hydroxyproline, then what you have is a collagen that denatures more readily. It has a lower melting temperature. So you can do an experiment where you take collagen and you can heat it up and see at what temperature is the helix half melted. So if I go from 0 to 1 and go to 0.5 and drop a line down, that will tell me the melting temperature. So this green curve right here shows collagen A, and here we just have a proline. Collagen B is shown here in blue, and that has a hydroxyproline, so you can see its melting temperature is a lot higher. Now this is not natural, but if you put a fluorine there, you get a very, very stable temperature. So again, this was done in the lab. Well, we don't naturally have fluorine as part of our fluorine is part of the proline. But what this tells us is that the reason that hydroxyproline makes our collagen more stable is because of that OH group. And the experiment with the fluorine, you might remember from organic chemistry that fluorine is strongly electron withdrawing. So it doesn't have anything to do with hydrogen bonding. I've highlighted in red to show that. It's tempting to think so because the OH is capable of hydrogen bonding, but that's not what it is. There are a couple of reasons why the electron withdrawal by the OH makes collagen more stable. The inductive effect might favor the trans orientation. Remember I told you earlier that peptide bonds involving proline might form cis double bonds, so maybe this helps the trans orientation. That's required of the peptide bonds in triple helical collagen. Or maybe hydroxyproline forms a puckered ring with the required phi and psi values where regular proline does not. So it has nothing to do with hydrogen bonding. The experiment with a fluorine shows us that. So this is why I was talking about the pirates, because if you don't have vitamin C, remember vitamin C was needed as a cofactor for the prolyl hydroxylase. So if you don't have vitamin C, the proline to hydroxyproline conversion doesn't occur. And we can see from that last experiment that you have collagen that's a lot less stable. And since collagen makes up skin and blood vessels and things like that, the symptoms are skin lesions, fragile blood vessels, loose teeth, bleeding gums. So those are all the symptoms you get as a result of fragile collagen. So they did discover this, that you might remember from your history that British sailors were given limes, and they discovered no scurvy if we give them limes. They didn't know why, but the limes, of course, contain vitamin C. So another thing that you might be interested in is that if you eat meat and you use meat tenderizer, it contains a collagenase, enzymes that cleave collagen. Because what happens as we get older is that here we have our triple helix. To make collagen even stronger, we can take some of these side chains, and maybe we take these lysines and convert them to, we can convert this to an aldehyde group to make allysine, and then we can do an aldol condensation. So now we have a covalent bond linking two triple helices together. So that happens as an animal gets older. So meat from older animals is going to be tougher than younger animals because this happens as the animal ages. But this again explains why meat tenderizer works, because you can cleave some of the peptide bonds in collagen and make the meat easier to chew and digest. So just a quick diagram of how we make collagen. So we show that the collagen is made in the rough endoplasmic reticulum. So we're making our chains right here, and then we will, once we've made our chains, we can hydroxylate them, and then once the chains are hydroxylated, we can assemble them to form a procollagen, and this will be secreted from the cell, and then, we are going here from the ER to the Golgi, and then we leave the cell. There are appendages on the the amino and the carboxyl terminus that guide the assembly of the triple helix, and we remove those. And then if each of those triple helixes is kind of like a brick, then we're going to use the bricks to build a wall and then do that cross-linking that I showed you to make a collagen fibril that forms the basis of the skin, blood vessels, and things like that. So that's our whirlwind tour through primary and secondary function of proteins. Thank you for your attention, and I will see you in class.